Usually when we talk about a medical diagnosis, there's this comforting expectation of precision. It feels almost um, like engineering.
Right, yeah. Like you fall off a bike, you break your arm, the doctor puts you in an X-ray machine.
Exactly. And a few minutes later, they pull up an image. There's this jagged white line on a black background, and the doctor just points to it and says, there it is. That's the problem.
It's entirely binary. Yeah, I mean, the bone is either broken or it's intact. We are so deeply conditioned to prefer that kind of visibility.
We really are. We want to be able to point directly to the source of the malfunction, but then you step out of the orthopedic clinic and, well, you step into the world of
Oh,
neuroscience.
absolutely. It's a totally different ballgame.
Yeah, you start looking at neurodevelopment or cognitive aging or how the brain actually metabolizes its fuel over the course of a human lifespan. And suddenly, that crisp, clean X-ray machine is completely useless.
It's essentially stepping into the dark. For a very long time, we simply didn't have the tools to measure how the brain powers itself on a moment-to-moment basis.
And because we couldn't see it, we fundamentally misunderstood some of the most critical maintenance systems keeping our neurons alive.
Right, which is exactly why we're here today. We find ourselves looking at a diagnostic landscape that is incredibly murky. It's not about a snapped bone anymore.
No, it's about microscopic shifts in energy, fading cellular signals and these cascades of invisible molecules.
Exactly.
Welcome to this custom-tailored deep dive created specifically for you, the listener. Our mission today is to completely deconstruct a biological concept that you probably thought you already had a firm grasp on.
We are going to uncover the secret life of estrogen.
And I use the word secret deliberately. I mean, for decades, the medical establishment and pop culture, for that matter, has boxed estrogen into this very neat, very contained little category.
Yeah, it's been labeled simply as a female reproductive hormone, and that's the end of the story.
But the actual story is just so much bigger than that. Today, we're exploring estrogen's hidden identity as a master regulator of brain bioenergetics.
When you look at the last 20 years of neuroendocrinology, a completely different picture emerges. I mean, we aren't just talking about mood swings or hot flashes.
Right.
We were talking about massive physical, structural impacts on how your brain produces ATP, which is the fundamental non-negotiable currency of all cellular energy.
We are going to see how estrogen literally rebuilds the physical structure of your brain cells from the inside out.
Yeah.
And how it actively wires the neural networks that allow you to, form a memory, solve a problem, or process an emotion.
The paradigm shift here is profound. Yeah. We're moving from viewing estrogen as a reproductive switch to viewing it as the foundational infrastructure of cognitive endurance.
And what blows my mind about the source material we're diving into today is the ultimate conclusion these researchers are reaching. Estrogen isn't just a fuel additive, it actually appears to be the ultimate biological shield against neurodegeneration.
It really is. We're going to explore how it acts as a literal defense mechanism against severe genetic mitochondrial diseases.
Understanding how this single molecule fuels the brain could completely shatter and rebuild the way we view cognitive decline, aging, and even Alzheimer's disease.
Absolutely. To guide you through this, we've assembled a massive stack of scientific literature. But, we aren't just going to read you a bibliography.
Right. We're going to trace a scientific mystery that has been unfolding across nearly 2 decades. We'll be looking at foundational breakthroughs, like the work done by Roberta Diaz-Brinton back in 2008.
It was published in Trends in Neurosciences, right?
Exactly. It really cracked the door open on how estrogen biases a cell toward health. We'll also pull from crucial turning points over the last decade, like Yao and Brinton's 2014 synthesis and advances in pharmacology.
And there's that phenomenal 2018 review by Ledgry, Grimm, and Eckert in Frontiers in Aging Neuroscience that mapped out the aging female brain.
Yeah, and we're weaving all of that historical context together with the absolute cutting edge of modern science. We have deep insights from Shaw's 2021 work in brain, behavior, and immunity health.
Plus, Guajardo Correa's 2022 research in Frontiers in Cell and Developmental Biology.
And finally, to bring this right up to the present day, we are grounding our conclusions in a sweeping 2025 review by Shi Huang and colleagues, published in Cellular and Molecular Neurobiology.
It is an incredible journey through the data.
So let's unpack this. Before we can even begin to talk about how estrogen is rewiring your memory networks or fighting off genetic diseases, we need to understand the playing field.
Right. We have to look at the brain's baseline energy demands.
And we need to figure out how a hormone that most people associate with the ovaries actually gets into the control room of the brain in the first place.
To start, you really have to visualize the sheer metabolic cost of human cognition. We take thinking for granted, but biological processing is incredibly expensive.
Let's look at the numbers laid out in that pivotal 2014 paper by Retberg and colleagues in Frontiers in Neuroendocrinology.
Yeah, they detail estrogen's role in bioenergetic systems, but they start with a stark mathematical reality. The average human brain makes up only about 2% of your total body mass.
So if you weigh 150 pounds, your brain is, what, maybe 3 pounds?
Exactly. Yet that 3-pound organ consumes 20% of the body's entire daily fuel supply.
Just pause on that for a second. 20% of your fuel for 2% of your weight. I mean, if you bought a car and a radio consumed 20% of the gas tank, you would think the engineering was completely flawed.
Yeah, it's a staggering ratio.
Why is this so greedy?
Well, neurons are essentially the most high-maintenance cells in the entire biological kingdom. Unlike muscle cells or fat cells, neurons do not store energy well.
Okay, so a muscle can store glycogen to use later when you decide to sprint, right?
Right. But a neuron cannot do that. It requires a constant, highly regulated, second-by-second delivery of glucose and oxygen to generate ATP.
And it needs that ATP not just to perform complex thoughts, but simply to exist, doesn't it?
Exactly. A massive portion of that energy goes toward maintaining the resting electrical potential of the cell membrane. The neuron is constantly pumping ions in and out, fighting against natural chemical gradients, just staying primed and ready to fire.
It's like a bowstring that is constantly pulled back, just waiting for the release. All that energy is spent just holding the tension.
That is a brilliant way to conceptualize it. Maintaining that tension takes constant ATP. Then add in the cost of actually firing an action potential, releasing neurotransmitters, and building new synaptic connections when you learn something.
So if there's even a fractional drop in that energy supply, the tension slips.
Yes, and neurons become incredibly vulnerable. They cannot just power down and wait for food. If they lose their energy supply, they begin to take on structural damage very quickly.
Okay, so the brain is this hyperactive, high-performance engine that is constantly burning fuel just to idle, let alone to accelerate. But where does estrogen fit into this picture?
This is where it gets really interesting.
Because, I mean, if I stop 100 people on the street and ask them where estrogen comes from, 99 of them are going to say the ovaries.
Oh, absolutely.
They associate it entirely with the reproductive system, the menstrual cycle, and pregnancy. So how is an ovarian hormone getting into the brain's engine room to manage energy?
This is the first major misconception we need to dismantle. The 2021 Shaw paper in Brain, Behavior, and Immunity Health addresses this beautifully.
Right.
Yes, in a female body, a massive volume of circulating estrogen is synthesized and released by the ovaries. But estrogen is not trapped in the bloodstream. Structurally, the estrogen molecule is lipophilic.
Meaning it loves fat, it dissolves in lipids.
Correct. And because the blood-brain barrier is essentially a highly selective lipid wall designed to keep toxins out of the brain, lipophilic molecules like estrogen can slip right through it.
Oh, wow.
Yeah, estrogen travels from the ovaries, flows through the systemic bloodstream, arrives at the brain, and just glides across the barrier without needing a special transport protein. It has unfettered access to the neural tissue.
I love that. It's like estrogen has an all-access VIP pass. It doesn't have to wait in the long line outside the club hoping the blood-brain barrier bouncers let it in. It just scrolls right past the velvet rope.
It does. But the story gets much stranger. The brain doesn't just sit around passively waiting for the ovaries to deliver this VIP molecule.
Wait, really?
Yeah, the brain actively manufactures its own estrogen entirely in-house.
I really want to make sure the listener is absorbing this. The brain is producing estrogen all by itself, regardless of what the ovaries are doing.
Both the male and the female brains synthesize estrogen locally. The neural tissue takes circulating cholesterol, or circulating androgens like testosterone, and converts them.
How does it do that?
Well, the brain is packed with an enzyme called aromatase. Aromatase's entire job is to grab testosterone and biochemically convert it into estradiol.
Which is the most potent and biologically active form of estrogen, right?
Exactly.
So even if you are a man, your brain is taking your testosterone and turning it into estrogen to run your neural networks.
Absolutely. This local production is vital. It ensures that estrogen is present right at the synaptic cleft, exactly where it is needed for fine-tuning memory and managing local metabolic spikes.
That's wild.
It isn't just a systemic hormone sloshing around. It is a locally printed, on-demand signaling molecule within the brain itself.
Okay, so the brain is printing its own VIP passes in the back room using aromatase. But I have to stop you here because the math still isn't working for me.
How so?
If the brain is so incredibly self-sufficient like, if it can just manufacture its own estrogen on demand from cholesterol or testosterone, then why is the transition into menopause such a massive neurological event?
Ah, that's the big question.
I mean, women report severe brain fog, memory issues, and exhaustion when their ovarian estrogen drops. If the brain has its own private aromatase factor, shouldn't it be totally fine? Why does it care what the ovaries are doing?
It comes down to the concept of the metabolic set point. The brain's internal production via aromatase is highly localized and very small in total volume. Think of it as a localized sprinkler system designed to water specific small patches of grass when they get dry. It is meant for fine-tuning.
Got it.
The ovaries, on the other hand, provide a massive systemic baseline. They are a torrential downpour that keeps the entire water table high.
So the brain's local supply is a squirt gun, and the ovaries are a fire hose.
A perfect distinction. The brain's metabolism calibrates itself to expect the fire hose. Over decades, the female brain sets its baseline energy consumption based on that massive flooding tide of estradiol provided by the reproductive system.
And then perimenopause hits.
Right. When perimenopause begins, the ovaries stop producing that massive volume. The fire hose gets shut off. The brain experiences a sudden, drastic drop in its total estrogenic environment.
But what about the local aromatase sprinklers?
They're still working, but they cannot possibly compensate for the loss of the entire systemic water table.
So the baseline expectation is suddenly violated.
Exactly. And because estrogen acts as the master key unlocking the brain's ability to pull glucose out of the blood and burn it for ATP, that sudden drop essentially pulls the rug out from under the brain's primary energy supply.
Wow.
The brain isn't just losing a reproductive signal, it is suddenly starving for fuel.
That completely reframes the entire experience of menopause. It isn't just an end to fertility, it is a systemic bioenergetic crisis. The brain is suddenly running on fumes.
That's exactly what's happening.
So now that we understand estrogen is inside the brain, whether it glided across the blood-brain barrier from the ovaries or was printed locally by aromatase, we need to look closer. We need to zoom in all the way down to the cellular level.
Right, down to the organelles.
I want to go down to the factory floor. Let's talk about the mitochondria, the microscopic power plants inside the neurons. What is estrogen actually doing when it encounters a mitochondrion?
To really visualize this, we have to look at a landmark 2008 paper published in Trends in Neurosciences by Roberta Diaz-Brenton. Her team essentially wanted to take a microscopic inventory.
What were they looking for?
They wanted to see the physical, structural changes that occur inside a mitochondrion when it is exposed to estrogen. To do this, they conducted a highly detailed proteomic analysis.
Now, for the listener who might not be a biochemist, let's break down proteomic analysis. What does that actually mean in the context of an experiment?
Think of proteomics as taking a complex machine, disassembling it completely, and laying every single gear, screw, and wire out on a table to catalog them.
Okay, I can picture that.
The researchers took brain mitochondria from female rats. Some of these rats had their ovaries removed to deplete their estrogen, and others were treated with 17-beta estradiol.
And then they just cracked the mitochondria open.
Basically, yeah. They extracted the mitochondria from the brain cells and used mass spectrometry to identify and count every single protein operating inside those microscopic power plants.
So they aren't just looking at the overall energy output, they are counting the actual physical pieces of machinery inside the cell.
Precisely. They were looking at the literal building blocks. Out of 499 specific mitochondrial proteins they detected, they found that adding estrogen actively upregulated 28 key proteins.
Upregulated meaning like it forced the cell to build more of them.
Yes. It didn't just tweak a chemical signal, it structurally increased the presence of specific enzymes required for energy production. It physically rebuilt the factory to be more efficient.
That is fascinating. It's not just telling the factory to work harder, it's literally installing more conveyor belts. What specific machines was estrogen building? Out of those 28 proteins, what were the heavy hitters?
The heavy hitters were the structural enzymes that drive the two main engines of mitochondrial energy production. First, the proteomics showed a massive upregulation of something called pyruvate dehydrogenase, or PDH.
Okay, what is the job of PDH?
TDH is the ultimate gatekeeper. When a cell takes in glucose from the blood, it breaks it down into a substance called pyruvate in the cytoplasm. But pyruvate can't just magically turn into energy.
Right, it has to get into the furnace.
Exactly. It has to be transported into the mitochondria and fed into the tricarboxylic acid cycle, the TCA cycle, sometimes called the Krebs cycle. PDH is the enzyme complex that sits the doorway, takes that pyruvate, and actively feeds it into the mitochondrial furnace.
So if the mitochondria is a coal-burning power plant, PDH is the shovel that scoops the coal into the fire, and estrogen is telling the cell to buy a lot more shovels.
That is exactly what it does. It widens the intake valve, ensuring that the fuel the cell gathers is actually utilized and not wasted. But the second major finding was even more critical.
What was it?
Estrogen significantly increased the expression of the ATP synthase F1 alpha and F1 beta subunits.
ATP synthase, the name implies it synthesizes ATP. What does this machine actually look like?
ATP synthase is arguably one of the most incredible structures in biology. It is the final step of the electron transport chain. It's an actual physical rotary motor embedded in the inner membrane of the mitochondria.
Wait, it's a physical motor?
Yes. As protons flow through it, the top part of the enzyme physically spins like a turbine. And as it spins, it jams molecules together to create ATP.
That's amazing.
By upregulating the specific F1 alpha and beta subunits, estrogen is fundamentally upgrading the structural capacity of the motor itself. It's installing more turbines to capture the energy.
Okay, I am picturing this microscopic turbine spinning furiously to create energy, but I'm stuck on the logistics of how this happens.
Where's the hang-up?
How does a hormone molecule floating around in the brain actually force a mitochondrion to build a new turbine? What is the chain of command there?
The chain of command has been a subject of intense debate, and the answers are laid out brilliantly in the 2022 paper by Guajardo Correa and colleagues in Frontiers in Cell and Developmental Biology.
What did they find?
They break down the dual nature of estrogen signaling, the genomic pathway versus the non-genomic pathway.
Let's start with the classic understanding, the genomic pathway.
Classically, endocrinologists viewed estrogen strictly as a nuclear hormone. In the genomic pathway, an estrogen molecule enters the cell and finds an estrogen receptor, or ER. The molecule binds to the receptor, creating a complex.
And then what?
That complex then physically travels all the way into the nucleus of the cell where your DNA is stored. It binds to the DNA itself and acts as a transcription factor.
So it's basically reading the genetic code and sending out a memo saying, the boss wants us to transcribe the genes for pyruvate dehydrogenase and ATP synthase. Start manufacturing.
Exactly. It's the classic corporate hierarchy. The executive order comes down from the corner office, the nucleus, and is sent down to factory floor.
That makes sense. That's the genomic pathway. And it works. It builds those 28 proteins. But there is a massive problem with relying on this pathway alone, isn't there?
A huge problem. It takes time. Gene transcription, assembling the mRNA, sending it out to the ribosomes, synthesizing the new proteins and folding them into functional shapes, that process takes hours, sometimes days.
And earlier, we established that neurons don't have hours. They need ATP on a second-by-second basis just to keep their electrical tension. If they have to wait 4 hours for the nucleus to print a new shovel, the cell is going to die.
Which brings us to the revolution in our understanding, the non-genomic pathway. What researchers discovered, and this completely shifted the paradigm, is that estrogen receptors are not just sitting around waiting to go to the nucleus.
Where else are they?
They are distributed throughout the cell, they are on the outer cell membrane, and remarkably, they are located physically inside the mitochondria themselves.
Wait, the receptors are inside the power plant.
Yes. We have to look at the 2021 Shaw paper here, because it highlights something extraordinary about a specific receptor called estrogen receptor beta, or ER beta.
What did Shaw find?
Shaw details experiments where researchers use a technique to pull the ATP synthase motor out of the mitochondrial membrane. And when they pulled the motor out, they found that ER beta came with it.
It was stuck to it.
It was physically part of the same complex. They're bound together. Yeah. The estrogen receptor is not just in the neighborhood. It is physically attached to the ATP-generating turbine.
Oh, wow.
So when estrogen enters the mitochondria and binds to ER beta, it doesn't send a memo to the nucleus. It directly, physically modulates the activity of the electron transport chain in real time.
Okay, this is where the story gets really, really interesting for me, because this totally changes the corporate metaphor we were just using.
Right.
ER-beta is not a factory floor manager who sits up in a glass office, typing out a memo, sending it to the nucleus, and waiting for the workers to build a new machine.
Not at all.
This manager actually walks down the stairs, steps onto the assembly line, rolls up their sleeves, grabs the gears of the ATP synthase turbine, and physically cranks the machine to make it spin faster.
That is a brilliant visualization. Estrogen is physically putting its hands on the machinery to increase output.
And if it's just physically touching the motor, how fast does this non-genomic reaction happen?
It is virtually instantaneous, because the receptor is already complexed with the enzymes on the mitochondrial membrane. Binding estrogen causes immediate conformational changes.
Like, how immediate?
We are talking about increases in ATP production and cellular calcium buffering within seconds to minutes, not hours.
So it is an immediate throttle response.
Yes, it provides an immediate, on-demand energetic boost to keep the neuron firing right this second, while simultaneously other estrogen molecules are triggering that genomic pathway, sending memos to the nucleus to build long-term capacity for tomorrow.
It operates on both timelines at once.
Exactly. Instant survival and long-term structural upgrades.
That dual action mechanism is incredible. It explains how a single molecule can be both a fast-acting neurotransmitter-like signal and a long-term developmental hormone.
It's remarkably efficient.
So if we look at a single neuron in a petri dish, we can see that estrogen is making it incredibly robust. It has more ATP, more shovels for the coal, faster turbines. But you and I are not single cells in a petri dish.
No, we are vast networks of cells.
Right. And to understand cognition, to understand memory and emotional regulation, we have to scale this up. We have to see how this microscopic seconds-fast ATP boost actually affects massive brain regions and the communication networks between them.
Scaling up is where the clinical reality of this biology becomes visible. Because in the brain, energy equals communication.
Okay.
If a specific brain region has more ATP, it can support more synaptic connections, it can fire more robustly, and it can send signals faster across the long axonal highways to other regions.
And how do we actually observe that macro-level effect?
To see this, we turn to the clinical neuroimaging data synthesized in Yao and Britton's 2014 paper in Advances in Pharmacology. They examined pivotal human studies, specifically longitudinal tracking done by researchers like Resnick in 1998, Maki and Resnick in 2000, and Razgan in 2005.
And these studies were using PT scans, correct? Positron emission tomography.
Yes, exactly.
Just to ground the listener, a PET scan isn't an X-ray. It doesn't show structure. In this context, it usually measures cerebral blood flow or glucose metabolism. Patients are injected with a tiny amount of radioactive glucose, and the scanner literally takes a picture of which parts of the brain are hungry and actively eating that fuel.
That is a perfect summary of the methodology. It's a real-time map of energy consumption. It shows us the metabolism in action.
So what did these specific studies look at?
In these studies, the researchers compared healthy postmenopausal women who were using estrogen-based hormone therapy against those who were not. They tracked them over time, and the visual and statistical results on those PT scans were striking.
What lit up on the scans?
The women using estrogen showed significantly higher, sustained regional cerebral blood flow and glucose metabolism in several key areas, specifically the hippocampus, the perihippocampal gyrus, and the temporal lobe.
Wow. For anyone who has studied basic psychology, those names should be ringing massive alarm bells. Those are the classic memory circuits. The hippocampus is the absolute center for forming new episodic memories and retrieving old ones.
Right. The memory centers were burning fuel brightly and efficiently. But crucially, the researchers also looked at a deeper structure called the posterior cingulate cortex.
The Razgan 2005 study was particularly revealing here, wasn't it?
Yes, it was. They found that women who were not receiving estrogen experienced a significant measurable decrease in metabolism in the posterior cingulate cortex over a 2-year period. The lights in that specific room were slowly dimming.
And the women on estrogen.
The estrogen users preserved their metabolic rate in that area.
Why does the posterior cingulate cortex matter so much?
Because a metabolic decline in the posterior cingulate cortex is widely considered to be one of the earliest known biomarkers of Alzheimer's disease.
Wait, really? The earliest?
Yes. The metabolism in this region begins to drop years, sometimes a decade or more, before any clinical memory loss actually appears. It is the canary in the coal mine for neurodegeneration.
Wow. So by maintaining the glucose metabolism in that specific area, estrogen is literally keeping the lights on in the exact tissue that is most vulnerable to the onset of Alzheimer's.
It is preserving the baseline infrastructure. But as you know, a brain region doesn't sit in isolation. The posterior cingulate cortex has to talk to the hippocampus. The hippocampus has to talk to the frontal lobes.
Right. The brain is a network, not a collection of islands. Does estrogen affect how these islands communicate?
Massively. Yao and Britton highlight functional MRI studies by Ottowitz from 2008, which looked specifically at network connectivity.
And what did they find?
They found that estrogen actively enhances the connections between the prefrontal cortex and the hippocampus, as well as the networks connecting the amygdala to the cortex.
Let's map those out. The prefrontal cortex is your executive suite. It handles decision-making, focus, complex planning. The hippocampus is your filing cabinet for memories. And the amygdala is your emotional processing center, handling fear, anxiety, and threat detection.
Right. And when you try to recall a specific fact to solve a complex problem at work, your prefrontal cortex has to send a signal down the highway to the hippocampus, retrieve the file, and bring it back up to your conscious awareness.
And if you're stressed, the amygdala is involved too.
Exactly. The amygdala is firing signals into that network. The Ottowitz data shows that estrogen makes these highways wider, faster, and more robust.
Let me see if I can picture this dynamically. Does this mean estrogen is acting as a sort of metabolic traffic cop?
That's a great way to look at it.
It's not just keeping the lights on in the buildings, it is standing in the intersection, keeping the massive highways of memory and emotional regulation clear of traffic jams. It ensures that when the prefrontal cortex needs to retrieve that memory file, the physical energetic bandwidth is actually there to make the call instantly, without static.
That is the functional result, yes. On a scan, a brain with optimal estrogen levels literally looks different in how it behaves. The network shows synchronous, robust activation.
Because the energy is there.
Precisely. The traffic flows smoothly because the underlying biological infrastructure, the ATP produced by those mitochondria down on the factory floor, is fully supported. The cells have the energy surplus required to maintain their synaptic connections.
And what happens if the energy surplus goes away?
The brain is ruthless about energy conservation. Without sufficient ATP, the brain begins to prune those synapses. It tears down the highways because it can no longer afford the metabolic cost of maintaining them. The communication degrades.
Which brings us to the terrifying part of the narrative. If estrogen is this incredible fuel injector, this traffic cop for the brain's massive networks, what physically happens when that supply plummets?
It's a drastic shift.
What is actually happening inside the cells of a woman going through the perimenopause transition?
To understand the cellular crisis of perimenopause, we have to dive deep into the 2018 paper by Ledry, Grimm, and Eckert in Frontiers in Aging Neuroscience. The paper is titled Mitochondria, Estrogen, and Female Brain Aging.
And they map out exactly what happens during this period.
Yes, they call it the critical period. When systemic estrogen levels from the ovaries drop, the brain experiences a sudden state of hypometabolism.
Hypometabolism, meaning the metabolic fire goes cold.
Exactly. The glucose transport proteins that estrogen usually keeps active start to shut down. The intake valves close. The mitochondria, suddenly lacking their factory floor manager cranking the gears, become sluggish and inefficient.
So the fuel lines get pinched off and the turbines slow down.
And when a mitochondrion becomes inefficient, two devastating things happen simultaneously. First, as we've discussed, ATP production crashes. The cell loses its currency.
And the second thing?
Second, and much more dangerous, the electron transport chain starts making mistakes. Because it isn't running smoothly, it starts leaking loose electrons into the cell.
Oh, that doesn't sound good.
It's not. These electrons bind with oxygen to create reactive oxygen species, or ROS.
ROS. These are the free radicals we hear about in skincare commercials, right? They cause oxidative stress.
Yes, but in a neuron, oxidative stress is not about getting wrinkles. Free radicals are highly unstable molecules that aggressively strip electrons from everything around them.
So they're just causing chaos inside the cell.
Complete chaos. They tear apart cell membranes, they shred cellular proteins, and they mutate mitochondrial DNA. The cell is literally being damaged from the inside out by its own power plant.
It sounds like an engine knocking. The car is out of tune, it's not burning the fuel cleanly, so instead of creating forward momentum, it just starts spewing toxic, superheated exhaust right into the engine compartment, melting the wire.
The toxic exhaust analogy is perfect. The neuron is drowning in ROS while starving for ATP. And because the brain is panicking over the lack of glucose, it tries to switch to emergency backup power.
What's the backup power?
The literary paper details how the hypometabolic brain starts upregulating genes for fatty acid oxidation. It shifts away from glucose and tries to rely on ketone bodies as a fuel source.
Now, people use keto diets all the time for brain clarity. What's wrong with burning ketones?
Ketones are a fantastic backup fuel. The problem is not the ketones themselves. The problem is where the brain sources the lipids to make them in this specific crisis state.
Where is it getting the fat?
In the aging, estrogen-deprived brain, chronic reliance on this alternative pathway can lead to the brain scavenging its own structural lipids. It begins breaking down myelin.
Myelin is the white matter. It's the protective insulation wrapped around the neural wires that allows signals to travel fast.
Precisely. The brain is so desperate for fuel that it starts stripping the insulation off its own communication wires just to feed the mitochondria.
It is literally tearing the walls off its own house and throwing the wood into the fireplace just to keep from freezing to death. That is a horrifying biological reality.
It's a severe crisis mode.
But listening to this, a massive, glaring question comes up. It brings up one of the biggest controversies in modern medicine.
I think I know where you're going with this.
If the drop in estrogen causes the brain to burn its own house down, why didn't hormone replacement therapy just fix everything? In the early 2000s, the Women's Health Initiative study famously terrified the public by suggesting HRT caused adverse health events in older women, including cognitive risks.
Right.
If estrogen is the ultimate mechanic, why didn't adding it back in just fix the engine?
You have hit on the most important paradigm-shifting concept in modern neuroendocrinology. The answer to the WHI paradox lies in a framework called the healthy cell bias hypothesis.
The healthy cell bias.
Yes. This was formulated by Roberta Diaz-Britton in 2008, and it aligns closely with the critical window hypothesis proposed by Barbara Sherwin in 2007. Both of these are thoroughly analyzed in the 2014 Yale paper.
What does that actually mean? Walk me through the mechanics of why giving an older woman estrogen might not help her brain.
The entire paradox hinges on the physical health of the mitochondria at the exact moment the estrogen is reintroduced. We have to remember how estrogen signals the cell to make energy.
Okay, remind us.
To activate those mitochondrial turbines, estrogen triggers a temporary controlled influx of calcium into the cell. Calcium acts as a signaling spark.
Okay, calcium is the spark plug.
In a healthy cell, one that is still relatively robust, the mitochondria can easily handle that calcium spark. They use it, buffer it, and turn it into massive ATP production.
So if estrogen is administered early enough?
Exactly. If it's given during the critical window, which is the phase of perimenopause or very early menopause, the neurons are still mostly healthy. The estrogen does its job. It manages the calcium, it sustains ATP, it clears out the toxic ROS exhaust. It acts as a shield.
It manages the factory beautifully because the factory is still structurally sound.
Exactly. But now consider a woman in the WHI study. The average age in that specific cohort was around 63. Many of these women had been fully postmenopausal, completely without estrogen, for 10 or 15 years.
So their brains had been running on fumes, accumulating toxic exhaust, and breaking down myelin for a decade.
Yes. Their neurological landscape had fundamentally changed. Their neurons had suffered years of chronic oxidative stress. The mitochondrial membranes were damaged.
Calcium.
Crucially, their ability to regulate intracellular calcium was completely broken. The cells were already constantly leaking and overwhelmed by toxic levels of calcium.
They were flooded.
So, into this diseased, overwhelmed cell, you suddenly introduce a massive dose of estrogen therapy. The estrogen does what it always does. It tries to send a calcium spark to turn on the engine.
But the cell is already flooded.
Right. The estrogen signal becomes the final straw. It pushes the calcium load past the biological tipping point, overloading the broken system and actually triggering apoptosis.
Apoptosis meaning programmed cell death. The cell just self-destructs.
It accelerates the degeneration. Adding the protective hormone to a diseased cell actually makes the damage worse.
I want to use a car analogy to make sure I'm visualizing this correctly. It's like pouring premium, high-octane racing fuel into a car. If you pour it into a well-maintained sports car, the healthy cell in the critical window, the engine roars to life. It performs at its absolute peak.
Yes.
But if you take that exact same high-octane fuel and pour it into an engine that has been sitting neglected in a junkyard for 10 years, an engine that is rusted solid, has degraded rubber seals, and is actively leaking oil, Pouring high-octane fuel into that rusted block isn't going to magically fix the engine. It's just going to cause a massive, catastrophic fire.
That is the healthy cell bias in a nutshell. Estrogen is not a magical healing potion that can resurrect dead tissue. It is an amplifier of cellular function.
So it amplifies whatever state the cell's already in.
Exactly. If the underlying function is healthy, estrogen amplifies health and energy. If the underlying function is severely compromised and diseased, estrogen amplifies the stress and accelerates the cell's demise.
That distinction is so critical for people to understand. It completely reframes hormone therapy. But I want to push back on the timeline here.
Okay, sure.
You mentioned the critical window. How narrow is this window, really? Are we talking about a few months? Because that would be terrifying for women trying to navigate the healthcare system to get treatment.
The exact timeline can vary based on individual genetics and health factors, but the scientific consensus, heavily supported by the Leisure 2018 review and clinical data from researchers like Lisa Mosconi, suggests the window aligns closely with the perimenopause transition itself and the early years of postmenopause.
So we aren't talking weeks, we're talking years.
Yes, roughly the 5 to 10 years surrounding the actual cessation of menses. It is a reasonable window of time. But the data is clear. Once a woman has been fully postmenopausal for a decade or more, the likelihood that her neural mitochondria have transitioned into that diseased or rusted state increases exponentially.
Wow.
This is why timing in hormone therapy is the single most critical variable. The goal is to preserve the bioenergetic infrastructure before it degrades, because once it has burned down, you cannot rebuild it from the ashes.
That is a profound shift in how we need to view the aging female brain. It's not just a transitional phase of life, it is a time-sensitive bioenergetic crisis requiring infrastructure preservation.
Exactly.
Which leads me to my next question. We know when estrogen is needed to protect the cell, but I want to know exactly how it protects the cell.
The specific mechanisms.
Right. When estrogen is present during that critical window, what specific defense mechanisms is it deploying to clean up the damage and prevent the engine from rusting out in the first place? What are its weapons against aging?
The intracellular defense mechanisms are breathtaking in their complexity.
Yeah.
The 2018 Leisure paper outlines these pathways in incredible detail.
What are the main players?
Well, estrogen doesn't just increase ATP output, it actively upregulates two massive, heavy-hitting neuroprotective players. The first is BDNF, and the second is SRT3.
Let's take them one at a time. BDNF stands for brain-derived neurotrophic factor. Anyone who listens to podcasts about neuroplasticity has probably heard of BDNF.
Right, it's very popular right now.
People casually call it miracle grow for the brain. We know it helps neurons grow and form new synapses. But how does BDNF relate to the mitochondria, the power plants?
The connection between estrogen, BDNF, and energy is a beautiful loop. Estrogen actively increases the expression of BDNF, and then both estrogen and BDNF converge to activate a master regulatory protein called PGC1 alpha.
PGC1 alpha, what is its job?
PGC1 alpha is the primary biological driver of mitochondrial biogenesis.
Biogenesis,
Right.
meaning the creation of new life. It builds new mitochondria.
Exactly. It literally tells the cell to synthesize brand new, highly functional mitochondria from scratch. It assesses the energy demand, sees that the factory needs more capacity, and builds entirely new factory wings.
That's incredible.
Estrogen ensures that the neuron is constantly repopulating its power grid with fresh, uncorrupted generators.
Okay, so BDNF and PGC1 alpha are the construction crew building new machines. But what about the second player you mentioned, SRT3? What does it do?
SIRT3, or sirtuin-3, is a fascinating protein. It belongs to a family of longevity genes. SIRT3 lives almost exclusively inside the mitochondria. Estrogen strongly upregulates the production of SIRT3.
And what does it do once it's inside?
Once SIRT3 is active, it acts as a molecular mechanic. It directly interacts with a potent antioxidant enzyme called manganese superoxide dismutase, or MNSOD.
MNSOD, that is a mouthful. What is its role?
MNSOD's only job is to hunt down and neutralize those toxic reactive oxygen species, the free radicals we talked about earlier. It scrubs the toxic exhaust.
But it needs SIRT3 to work.
Yes. MNSOD doesn't work at full capacity on its own and needs to be activated. SIRT3 performs a chemical process called deacetylation on the MNSOD molecule.
Decetylation, visualize that for me. What is SIRT3 actually doing to the molecule?
Think of the MNSOD antioxidant as a fire extinguisher, but it has a safety pin stuck in the handle. Decetylation is the physical act of pulling that safety pin out.
I see.
SIRT3 pulls the pin. Once deacetylated, the MSOD is supercharged. It rapidly, aggressively neutralizes the oxidative stress, turning the toxic free radicals into harmless water.
Wow.
By upregulating SIRT3, estrogen ensures that the mitochondrial engine is constantly being scrubbed clean of its own toxic exhaust.
That is an incredibly elegant defense system. Estrogen uses PGC1 alpha to build brand new power plants, and it uses SIRT3 to pull the pin on the fire extinguishers to keep the existing power plants clean.
It's a two-pronged approach, yes.
But biology is messy. What happens to the mitochondria that are just completely broken? The ones that are so damaged by age or stress that SIRT3 can't fix them, and they are just sitting there leaking toxic exhaust everywhere.
That is the ultimate test of cellular survival. And to answer it, we turn to the absolute latest findings. The 2025 review by Shi Huang and colleagues in cellular and molecular neurobiology zeroes in on exactly this problem.
What do they look at?
This paper delves deep into how estrogen regulates a process called mitophagy.
Mitophagy. Now, I know autophagy is the process where a cell cleans up its general waste. Is mitophagy just autophagy for mitochondria?
Yes, it is a highly specialized, targeted garbage disposal system designed specifically to identify, swallow up, and dismantle dead or severely malfunctioning mitochondria. It is the ultimate quality control mechanism.
And estrogen controls this. How?
The 2025 Huang paper maps out the exact circuitry. Estrogen regulates mitophagy through two primary sensing and signaling pathways. First, it activates the AMPK pathway.
What is AMPK?
Think of AMPK as the cell's internal fuel gauge and damage sensor. When a mitochondrion stops producing ATP and starts leaking toxins, AMPK sounds the alarm.
So it flags the broken machine.
Once the machine is flagged, estrogen utilizes the PI3K-AKT pathway to promote the formation of something called a mitophagosome.
Mitophagosome, let me guess, that's the biological garbage bag.
That is exactly what it is. A double membrane structure forms, physically engulfs the broken mitochondrion, seals it shut, and then delivers it to lysosomes, which are essentially vats of acid that dissolve the broken machine down to its basic amino acids so the parts can be recycled.
That is so cool.
Estrogen acts as the supreme quality control inspector on the factory floor. It walks the line, and if a machine is irreparably broken, it flags it, bags it, and melts it down before it can poison the rest of the neuron.
I cannot help but picture this as a microscopic cellular Marie Kondo routine. The estrogen inspector walks into the cell, looks at a sputtering, leaking mitochondrion, and asks, does this spark joy?
Right. Or in metabolic terms, does this maintain its membrane potential and produce ATP?
Exactly. And if the answer is no, estrogen helps the cell politely thank the mitochondrion for its years of service, shoves it into a garbage bag, and completely obliterates it.
It is a ruthless but entirely necessary form of biological tidying up.
Which forces me to look at the dark side of this equation again. If I follow the logic, if a brain loses its estrogen supply and it doesn't have HRT, the Marie Kondo inspector retires.
The PI3K and AMPK pathways stall.
And the cell loses its ability to clear the trash? Does the neuron basically become a biological hoarding house for dead, rotting mitochondria?
Tragically, yes. Without estrogen driving the mitophagy process, the dead mitochondria physically pile up inside the cell body. They take up space, they fail to produce any usable energy, and they leak massive, unchecked amounts of oxidative stress into the cytoplasm.
That sounds disastrous.
It is. This hoarding scenario, the accumulation of damaged toxic mitochondria, is not just a symptom of aging. It is a primary driving hallmark of almost every major neurodegenerative disease we face, including Alzheimer's, Parkinson's, and Huntington's disease. The inability to take out the cellular trash is fatal.
That paints such a vivid, terrifying picture of neurodegeneration, but it makes the bioenergetic argument crystal clear. Estrogen is the maintenance screw.
It really is.
So, we have talked extensively about normal, healthy aging, the menopausal transition, and how these microscopic defense mechanisms like SART3 scrubbing exhaust and mitophagy bagging up the trash protect us. Now, I want to take all of this machinery and apply it to a worst-case scenario.
Okay, let's do it.
What happens when these defense mechanisms are put to the ultimate test? Specifically, what happens when a patient is born with a genetic disease that fundamentally breaks their mitochondria from day one?
To explore that extreme scenario, we look again at the 2025 review by Huang and colleagues, focusing on their section on real-world clinical impact.
Which diseases do they look at?
The researchers examine severe genetic mitochondrial diseases, often abbreviated as MDs. They look at diseases like MELES, which causes encephalomyopathy and stroke-like episodes, and a particularly devastating disease called LHON, or labor hereditary optic neuropathy.
Let's focus on LHON. These diseases are caused by actual mutations in the mitochondrial DNA, right?
Yeah.
It's not just aging. The blueprint for the factory is fundamentally flawed from the moment the person is conceived.
Exactly. The genetic code required to build the ATP synthase turbines and the electron transport chain is mutated. And the clinical data surrounding LHON provides perhaps the most stunning, undeniable evidence of estrogen's sheer bioenergetic power.
Describe the disease for us.
LHON primarily attacks the retinal ganglion cells, the neurons that form the optic nerve. It causes rapid, painless, and severe loss of central vision. It usually strikes young adults in the prime of their lives, leading to permanent blindness.
That is heartbreaking, but how is the disease passed down?
The genetic mutation is located entirely in the mitochondrial DNA. And because you inherit all of your mitochondria directly from your mother's egg, LHON is maternally inherited.
Oh, I see.
This means that a mother carrying the mutation will pass it down to all of her children. Both her sons and her daughters will inherit the exact same mutated mitochondrial DNA.
Okay, so a brother and a sister both have the exact same flawed blueprint, the same broken factory.
Yes. But here is the extraordinary twist that puzzled researchers for decades. There is a stark, undeniable sexual dimorphism in a HOM, who actually develops the disease and goes blind.
What do you mean?
According to the data compiled in the Huang paper, males have a significantly higher incidence rate. A man with the mutation is anywhere from 2.92 to 4.8 times more likely to actually go blind than a woman with the exact same mutation.
Almost 5 times the risk for men.
Furthermore, males experience a much earlier onset of the blindness. The median age for men to lose their sight is between 20 and 25 years old.
And the women.
For the rare women who do express the disease, the onset is significantly delayed, usually not hitting until ages 28.5 to 33, or sometimes much later.
I need to wrap my head around this. They have the exact same maternal genetic mutation, the exact same broken mitochondria in their optic nerves. Why are the young men going blind at 5 times the rate, and doing so nearly a decade earlier than their sisters?
The scientific community has concluded that it is almost entirely due to the protective effects of estrogen. The Huang paper synthesizes the exact molecular mechanisms of how this works.
How does it work?
The specific genetic mutation in LHON causes complex I of the electron transport chain to be highly inefficient.
Meaning the turbine is broken and leaking.
Yes. It leads to a massive chronic buildup of oxidative stress and a severe deficit in ATP in those delicate optic nerve cells. The cells simply run out of energy and die, severing the connection between the eye and the brain.
So what's happening with the women?
In females, their naturally high circulating levels of systemic estrogen provide a profound energetic and antioxidant buffer that the males simply do not have.
Estrogen acts as a biological shield against their own genetics.
A literal, measurable shield. Estrogen crosses into those optic nerve cells. It binds to the ER-beta receptors physically located inside the mutated mitochondria.
And it forces them to work.
Yes. It forcibly upregulates whatever functional ATP synthase is left, squeezing every possible drop of energy out of the broken system. It drives SIRT3 to activate MNSOD to aggressively scrub the massive amounts of reactive oxygen species caused by the mutation.
Wow.
And it promotes massive mitochondrial biogenesis through PGC1 alpha, trying to overwhelm the mutation with sheer numbers of new mitochondria.
It's like the cell is a sinking ship with a hole in the hull, and estrogen organizes a massive crew with buckets to bail the water out faster than it can flood in.
That is exactly what happens. Estrogen forces the defective mitochondria to work so efficiently and cleans up their mess so thoroughly that it actively masks the underlying genetic flaw.
That's incredible.
The disease there, the genetic mutation is absolutely there, but the clinical penetrance, the actual blindness, is drastically reduced because estrogen simply will not allow those optic nerve cells to die. It fights the genetics with sheer bioenergetic force.
That is awe-inspiring biology.
It really is. It is only when the overall disease burden becomes overwhelmingly high, or if the female's estrogen levels fluctuate or drop, that the biological shield might slip, allowing the blindness to express.
But the men don't have that shield.
Right. For the young men lacking that massive systemic estrogen shield, the oxidative stress from the mutation kills the optic nerve cells rapidly, usually right as they reach their early 20s.
That is genuinely one of the most incredible things I have ever learned about human biology. The idea that a single hormone can essentially hold back the tide of a genetic destiny. But this realization immediately makes me look at the clinical reality.
The treatment side.
Yeah, I have to ask you about the treatment protocol here. If we know definitively that estrogen is the shield protecting these patients from going blind, Why on earth aren't we actively treating these young men with estrogen the moment we realize they carry the LHON mutation? Why let them go blind if we have the biological key?
It is a brilliant question, and it highlights the friction between cellular biology and whole-body medicine.
Yeah.
The reality is that treating young men with the massive high doses of systemic 17-beta estradiol required to protect the optic nerve would indeed prevent the neuropathy.
It would save their sight.
It would, but it would also cause profound systemic feminizing side effects.
Right. Gynecomastia, changes in body fat distribution, complete loss of libido, potential emotional changes.
Exactly. For most men, those side effects make systemic estrogen therapy clinically unviable and psychologically devastating. You are curing the blindness, but fundamentally altering their endocrine identity.
Which is a massive trade-off.
It is. However, this exact dilemma is what is driving the absolute bleeding edge of modern neuroendocrinology. The goal of the field right now is to separate the metabolic benefits of estrogen from the reproductive side effects.
How do you separate them if it's the exact same molecule?
You don't use the natural molecule, you design a new one. Researchers are actively developing what are known as SERMs selective estrogen receptor modulators.
I've heard of those.
And taking it a step further, they are developing phytoserms, which are plant-derived, and specifically mitochondrial-targeted synthetic analogs.
Designer molecules built in a lab.
Yes. The goal is to bioengineer a synthetic molecule that perfectly mimics estrogen, but with a highly specific structural shape. We want a molecule that perfectly fits into the estrogen receptor beta, the receptor located down on the mitochondrial factory floor.
Okay, so it fits into the turbine manager.
Exactly. It binds there, turns on the ATP turbines, activates the RT3 fire extinguishers, and protects the cell. But crucially, the designer molecule is shaped so that it cannot bind to estrogen receptor alpha, which is the receptor predominantly found in breast tissue and the reproductive organs.
So you get the bioenergetic shield, the ATP boost, the clean mitochondria, but absolutely zero feminizing effects on the rest of the body. You get the metabolic key without the reproductive baggage.
That is the holy grail of neuroendocrinology right now.
Okay, we need to pull all of this together because the sheer scale of the ground we have covered today is monumental. We started by tearing down the old textbook definition of estrogen.
We really did.
We learned it isn't just an ovarian hormone, but a lipophilic VIP molecule that glides effortlessly past the blood-brain barrier. We learned the brain even prints its own local supply using aromatase, all just to feed the staggering 20% energy demand of our neural tissue.
And then we zoomed all the way down to the factory floor of the mitochondria.
Ugh.
We saw how the estrogen receptor isn't just sitting in an office sending memos, but is physically bound to the ATP synthase motor, cranking the gears to produce energy in a matter of seconds.
Right, and we watched it widen the intake valves to burn fuel more efficiently.
Then we scaled back up to look at human PET scans, watching the hippocampus and the posterior cingulate cortex light up.
We saw estrogen acting as a metabolic traffic cop, keeping the structural highways between our logic centers and our memory centers clear of traffic jams, staving off the earliest markers of Alzheimer's.
We navigated the perilous biological crisis of perimenopause. We unpacked the WHI paradox, realizing that estrogen is high-octane racing fuel that absolutely requires a healthy, non-rusted engine to work safely.
We defined the critical window where intervention can actually save the cellular infrastructure.
We watched estrogen coordinate a masterful defense system, building new power plants with PGC-1 alpha, pulling the pins on antioxidant fire extinguishers with ERC-3.
And acting as a cellular Marie Kondo, using mitophagy to ruthlessly hunt down and discard dead mitochondria before they turn the neuron into a toxic hoarding nightmare.
Absolutely.
And finally, we saw the ultimate proof of its power in the real world. Estrogen acting as an impenetrable biological shield, masking severe genetic mutations like LHON, and saving women from blindness by overpowering defective DNA with sheer brute bioenergetic force.
It's an incredible story. And synthesizing all of that leads us to a paradigm-shifting final thought for the listener. Throughout this entire deep dive, we have methodically redefined estrogen.
Yes, we have.
We have stripped away its label as merely a sex hormone and revealed it to be a fundamental, ancient regulator of cellular bioenergetics. This realization gives birth to a new frontier in medicine, the concept of mitoceuticals.
Mitoceuticals, medicines designed specifically for the mitochondria.
Exactly. If estrogen's true underlying superpower in the brain is energy regulation and infrastructure defense, then the future of neuroscience relies on permanently untethering this incredible molecule from its reproductive legacy.
Right.
Imagine a world in the very near future where we successfully synthesize and deploy those highly targeted, non-feminizing estrogen analogs we discussed.
The designer keys.
Yes, in that world, we could theoretically biohack brain energy to cure cognitive decline.
Mm.
We could treat genetic mitochondrial diseases, halt the progression of Parkinson's, and perhaps even prevent Alzheimer's in aging populations, entirely regardless of their biological sex.
That is just
We could give the aging male brain, or the postmenopausal female brain, the exact metabolic shield it needs to survive, without triggering a single systemic side effect. It shifts our entire approach to aging, from managing symptoms to literally sustaining the light of consciousness at the cellular level.
That is an incredibly hopeful, brilliant vision for the future of medicine. It completely changes the narrative of what is possible. Thank you to you, our listener, for coming along on this deep dive into the source material. We hope we shattered a few of your preconceptions today and provided you with a powerful new lens through which to view your own biology. Stay insanely curious. And the next time you think about your brain or you hear a diagnosis, forget about that neat, clean binary X-ray showing a simple broken bone. Picture instead a vast, shimmering, highly active metabolic highway, constantly humming with electrical tension, continuously being paved, cleaned, and fiercely protected by the single most misunderstood molecule in the human body.